So first thing is the website's new to me too. Several of you had trouble. You've got access code and it's asking you for some kind of course code or something. So don't panic. If you managed to get access to the website, great. If you haven't, just give me a couple days to work through that. It's brand new to me too. It appears that I have to go on and create a class. So I'm learning. We'll do that. If you can get onto the website without being in the class, have any of you been successful doing that? No? Okay. A couple people. Then you see all the resources available. And most of you have been in lab already. So you're talking about stuff that you're going to do in lab, potentially using these resources. Okay. New products. Work through it over the course of the semester. In the meantime, what happens in here is of critical importance. So during the last class meeting, I mentioned to you that there are 4 really big, huge, important inorganic ions: sodium, potassium, calcium, chloride. And they're just going to come up over and over again in this class, right through to the end of the term in the spring. Today, I'm going to talk about organic macromolecules, and the 2 of those are going to be tied together. So your textbook and the images here, the stuff you have available online, any A&T book is going to go through these things. I'm going to do it a little different than any of them. So my presentation to you about the inorganic and organic components here, the ions, the charged ions, and then these macromolecules, is going to be a woven story together. I intend for you guys to be able to answer the question not only what is it, what does it look like, but the more important question is, why should I care, right? What does this mean to anatomy and physiology? So I'm going to do that with you, talking about that. So I've drawn a little diagram here today that's going to come up. A couple of the really important organic macromolecules are drawn on this diagram. This is intended to be a oversimplified picture of a cell membrane. All right? So I'll just label it here. Now, these little dots with the 2 lines drawn here on the cell membrane are intended to represent a particular kind of lipid that I'm going to talk about today. I'm hoping most of you at some point have been in your life been introduced to this. This is the famous phospholipid bilayer of the cell membrane. So we're going to talk about it when we get to the lipids. All right? This circle that I've drawn in the cell membrane is intended to represent a protein. That's also going to be one of the organic macromolecules I'm going to talk about today. Proteins. And the reason I've drawn it here is because during the first class session I told you about important charged ions. And I want to bring up 2 of them already in this class. And they are sodium and potassium. So if this represents the inside of the cell and this represents the outside of the cell, one could make the case that the human body does not work. You will cease to function as a human being if you do not have an unequal distribution of sodium and potassium ions around your cell membranes. You must have, outside the cell membranes, a high concentration of sodium, and you must have inside the cells, in the cytoplasm of the cells, a high concentration of potassium ions. The unequal distribution of these 2 inorganic ions will be the foundation of everything I teach you in physiology. Now, here's its context for today. Today you're going to learn about some organic macromolecules. We're going to start with the sugars, and then we'll work through the lipids and the proteins and the nucleic acids. I talk about basic structure. But the real question behind all this is going to be, why should I care? What does it matter if I know the structures of these molecules? Okay, so I'm going to start with context, then I'm going to teach you the structures, and I'm going to throw context in every one of them as we go. Okay, so this one ties the inorganic pieces together. Now, the cell membrane is in fact a very nonpolar or uncharged structure. The portions, the internal portions of this membrane do not have charge. And if you have something that carries charge, like sodium or potassium, the non-charged or non-polar portions of the membrane will resist those things. So you already know that there's more sodium outside than inside, and there's more potassium inside than outside. This you know. So the first question you should be scratching your head over is, how did that happen? If it's so important for life, how did it happen? So I'm going to answer that question today when we get to the proteins. How did this happen? The second question is, after it's actually happened, why does it matter that it happened? And the answer to that question is, there is nothing in your nervous system or in your muscles, both content for this first anatomy and physiology class. There's nothing about nerves or muscles that will work if this is not in place. You must have an unequal distribution of these ions for your nervous system to work. You must have an unequal distribution of ions for muscles to contract. They will not work otherwise. So you're going to learn a lot of details about this over the course of the semester. But for today, I want you to see the answer to the question, why does it matter? How does it work? What does it matter that I know these things? This is the why question. So in order for a nerve to work, in order for a muscle to work, sodium and potassium must cross the cell membrane. And if the cell membrane is not charged, and these ions are charged, it creates a barrier that is impenetrable. You cannot get them across the cell. And if you cannot get them across the cell, you cannot work. Your body won't work. So how does that happen? And the answer is, there are proteins. So now you've been introduced to the lipid and the proteins. There are proteins within the cell membranes that will allow these ions to pass through them. So how does sodium get across the cell membrane? The answer is, sodium passes through proteins. How does potassium get across the cell membrane? And the answer is, potassium passes through proteins. Now, if this doesn't happen, your brain doesn't work. If this doesn't happen, you don't get signals sent to muscles. And if this doesn't happen, your muscles will not contract. So I want you to see the importance of learning the structures of these organic macromolecules as we go. So you're already clued in to the lipids and the proteins in relationship to the charged ions that I brought up last time. Now, let's go through these, and I'll throw in functional pieces for this as we go through each of the 4 organic macromolecules. So sugars first. We call the sugars the carbohydrates. Let me throw the question out there. If I ask you, and certainly there's no wrong answers, I don't want you guys to feel intimidated to answer these questions. So if I ask you, what is sugar for in the human body, how would you answer that question? What do you need sugar for?
Sugars are converted to starches for energy.
Okay, great. So it's exactly what we're about to look at. So a starch is in fact a large sugar molecule. And whenever your body needs energy, one way of making that energy is to cut the smaller pieces of the sugars off and use them to make energy in the form of ATP. So that's a good way of thinking about sugars. We need them for energy. Now, we're going to see some more things that sugars do as we progress through this class. For example, as we're talking about cell recognition stuff, the semester wears on, you're going to learn that a lot of cells have sugar pieces on the outsides of the membranes that help cells recognize each other. You're also going to learn when we get to the cardiovascular system that there is this sugar-laden coat along the inside walls of your blood vessels that has some really important functions. Like one of them is it keeps the platelets from sticking and making blood clots. So sugars do a lot of important things. For today, I want you to learn the basic monomers, putting together simple monomers to make disaccharides, and then starch and glycogen as the polymer. So put it together in sequence. The first part of the sequence is, what are the important monosaccharides, the simplest sugar units in the human body. There are 5 of them that are important for human physiology, and they're on the board for you: glucose, fructose, galactose, deoxyribose, and ribose. Now, you should put these in your mind. For the purposes of this class, you should put them in 2 groups. The first 3, glucose, fructose, and galactose, are 6-carbon sugars. The sugars are made up of 6 carbons. Now, I will not expect you, because it doesn't have any relevance yet for us, I will not expect you to be able to draw these. So if you can remember that the 3 6-carbon units are glucose, fructose, and galactose, I'm okay. Those are the 6-carbon units. And, as this young man said, when you see those, the first thing you should think is energy. So in the opening chapters of your book, different in chapter 2 or 3 or 4, somewhere at the beginning, one of the major concepts that is covered a lot of times in A&P classes is where energy comes from. Now today I'm going to show you the structure of adenosine triphosphate, because that's the energy molecule for the human body. But if you want to begin a discussion about making energy for the human body, this is where the book will start. With glucose. Breaking down glucose to make energy. So that's the first thing that should come to your mind. Now, the other group of monosaccharides, our simple sugar units, are the 5-carbon sugars, and those are deoxyribose and ribose. These 2 monosaccharides are sugars that are used to build nucleic acids, and we haven't looked at them yet. So these 5-carbon simple sugars, deoxyribose and ribose, I'm going to put them in just a little bit. I'm going to put them on another organic macromolecule for you. But you shouldn't think energy when you see those. When you see those 2, you should think DNA and RNA. So I'm going to show you that structure here in just a minute and talk about those a little bit more. Okay, let's put them together and make some simple disaccharides. So this is a memorization thing, and it's going to introduce to you basic chemistry. Now, this is not a chemistry class. There is chemistry associated with opening chapters in every A&P book. I'm not teaching you ionic and covalent bonding and stuff like that. I have way too much to do to go through all that with you. So I'm assuming that you've had some chemistry. I'm not asking you about it. If you've got questions about these things, certainly I can help, but it's not the point of this class to teach you all the details about how these things are bonding and put together. However, as we talk about energy production later in the class, for example, instead of teaching you the real sophisticated energy mechanism, the glycolytic pathway and the Krebs cycle and oxidative phosphorylation, instead of doing that now, I'm going to wait. And after I have taught you muscle physiology, here's how a muscle contracts. After I've taught you that, then I'm going to say, in order for the muscle to contract, it has to have energy. And so the question we need to answer is, where does this energy come from? So one of the things we're going to have to discuss at that point is how you break these sugar molecules down to make energy out of them. All right? So you need to know the basic terminology for that. Okay? So first on the list here, you can see that glucose as a monosaccharide added to fructose as a monosaccharide forms a sugar disaccharide called sucrose. Very famous sugar, this table sugar. This is stuff that you put in your tea, sucrose. It's a disaccharide. The mechanism for making this disaccharide, and for that matter, for making a polysaccharide like glycogen or starch, that process is called dehydration synthesis. A water molecule is removed in the construction of organic macromolecules. This is a common phenomenon. First time I've said it today, but I'm going to say it with all of them. It's going to come up over and over again. If you want to build an organic macromolecule, the process is called dehydration synthesis.